So welcome everyone to this Physics of Life seminar, which will be given by Jose Onuchic from Rice University. And welcome everyone for being here. So before we start, again, as usually, mute your microphones. I think everyone is muted anyway, so there's no interference. Please submit questions in the chat box. We might ask some during the talk, and more we can ask after the talk. And we might also ask you to unmute then yourself. Otherwise, note we record. Okay, so it's my great pleasure to introduce Professor Jose Onuchic. He's a Harry C. and Olga K. Vice Chair of Physics, of Chemistry and Biosciences, and he's also the co-director of the NSF-sponsored Center for Theoretical Biological Physics. He won numerous prizes and awards, I mean, really a lot, so I can only mention a couple of them. So he won the ICTP Prize in honor of Heisenberg in Trieste in Italy, and the Beckman Young Investigator Award. I think that was already a while ago. And now he's a fellow of many societies, including the APS and the Biophysical Society. And he is also a member of the US National Academy of Sciences and also the Brazilian Academy of Sciences. So, so briefly about the CV. So Jose studied at the University of Sao Paulo and obtained a PhD from Caltech, where he also did a postdoc with John Hopfield. And he spent also some time at Santa Barbara and then became faculty at UC San Diego. And 2011, I think he went to Rice University. So Jose is very well known for a lot of things. So I remember he coins this folding funnel hypothesis for essentially solving, I think, the Leventhal's paradox for protein folding. And he worked also on charge transfer in proteins and later genetic networks. And I think now at Rice, he switched more or less to biomedical sciences, in particular cancer, if I remember correctly. But he's also a very fantastic teacher. I remember him from about 20 years ago from a summer school in Rhode Island. That was a fantastic summer school. And then also a little later from a seminar, he gave a biophysics seminar at Princeton University, you know, the one Bill Bialik runs. Yeah, and then, yeah, I'm really excited that he's here and that we hear his talk today. And the title of his talk, I think, is Energy Landscapes, Order and Disorder in Protein Sequence Coevolution. So over to you, Jose. Thank you for being here.
Well, thank you very much for inviting me. It's great to be here. Basically, I think Imperial has one of the great programs now putting together on the idea of what I call this sort of physics and life science coming together, and the program called Physics of Life. So it's good to be telling you a little bit about it. So I like this topic because a real topic or something that how do you get something that you know just from physics and has an enormous application into biology, protein folding, and many other areas. And this idea of coevolution is something that I started in 2009, and it's very popular now, as you're going to see as I talk about it. But let me start on the topic and move forward. So just to understand a couple things here to make sure, if there's any question, you ask me, right? Because I'm not reading the chat. So feel free to.
Yeah, you don't have to monitor the chat. We look at it and we might ask you once in a while a question.
Okay, so feel free. And you wanted me to talk for about 40 minutes, right?
Yes, that's right, yeah.
So I will try to stay on that time, not going to abuse you too much. Okay, so as we discussed here, the idea of the funnel concept is the things that basically we did for a long time, that when proteins fold, you have something like, for example, the system orders at the same time as the energy of the system come down. Here is effective energy already with solvation. So you have many paths come down to folding, and that means that this relationship between order and energy tells you that by knowing the ground state of the protein, you can learn a lot about the excited states of the protein. So because of that, with this idea of the folding hypothesis of the funnel, that basically pretty much telling you that when proteins fold, native interactions tend to be attractive and non-natives tend to be repulsive. Not 100%. That's sort of, in general, that allow the idea of what we call structural-based models. These are models that you know the structure of the protein. You can do a model using exactly this idea that native interactions are attractive. and non-natives are repulsive. And you can learn a lot about the proteins, not only about folding, but about functional aspects and situations like that. The problem with that is that in order to build these models, you have to know the structure. And we learn a lot about structures from people doing from X-rays, NMR, and other techniques these days with cryo-EM. You're getting a lot of structures, but it's still very limited. So the question becomes, how can I extend this idea towards situations that have limited information about structure? And one of the things came with this idea of coevolution. And coevolution, we're very proud because that's the main reason of success for AlphaFold. Everybody's talking about it these days. They're talking about AlphaFold, and really basically they use our ideas of coevolution, but they do much better data sets. But I'm glad that this idea that we proposed sort of 11 years ago is now become mainline on protein structure prediction by the engineering aspect of it. So this idea comes from the fact that if you look all the structures of proteins that you know, you're going to observe that you have many more sequences than you have structures. So for example, here's an example of a single folding motif, a helix turn helix folding motif. And you can see it appears at many different stages of evolution and many different organisms, but they have the same structure. So the question is, can we now do some statistics about these different sequences that have the same structure and come to some idea of what the structure is by just looking at sequence information? So the concept of coevolution comes from the following fact. As I told you before, I said that native interactions tend to be attractive and non-native interactions tend to be repulsive. If I take this idea at heart, you're going to see if I have an interaction that's a native interaction in a protein like I'm showing here, if I mutate one of the residues, I'm going to have to compensate that mutation by another residue to make it attractive. So if you assume that this hypothesis is happening on the different sequences that fold into the same structure, the base interactions are attractive, they have to be compensated. What you observe from here is that now you don't look at single residues, but you look at pair of residues. So for example, you look at this particular pair of residues and observe that this RE changes for ML. So you look at how residues vary together. So instead of looking at variance, you look at this, instead of looking just the conservation of residues, you look at this covariance of residues that appear into you. So this idea tells us now that we can try to use this information to predict and find native interactions into proteins. And if I know this native interaction, I can know about the structure. Now, why the physics become very cool here? Because you can observe the way is what's the input data to try to get information. Okay, first of all, you're going to try to get residue conservation. So I look, for example, multiple sequences that follow the same structure. Here I'm just showing this cartoon here. I sort of align them. Actually, this method became very powerful these days because alignment became much better, and also people start with the concept of protein families, so everything went better. So you align all the sequence. Then you can observe for each position on sequence which amino acid exists, what's the percent of occupation for each amino acid, and you get this quantity Fi. And the second quantity you have to get is the pair probability distribution. That's what, if you look positions, two positions, what's the probability you have two amino acids? So for example, if I look at this particular sequence, I have a line here, I'm looking position 6 and 17, and I say, what's the probability of having the pair S and P? And it's going to be 2, 6. So what we do is that statistics for lots and lots and lots of sequences. This information here is sufficient to give you mutual information, but they are not sufficient to give the probability of two residues are in direct contact, because residues may interact directly or indirectly. In order to get them direct, you have to get a global probability distribution. And to get a global probability distribution, first of all, we have this quantity, we get my covariance matrix. I can get this term for the global probability distribution that comes, yes? Yeah. So you get this term for global probability distribution, but what is very interesting, you observe that this term is just like a Potts Hamiltonian. where this term here is just to come from the conservations like the field term, and here is the interaction term. So now I can use all the tools that I know from magnetism and Potts Hamiltonian to apply to that problem, right? Now, so the complication about getting global problem distribution, if you have to do an inference, where I say which are the best parameters that I call here EIJ that satisfy all the constraints that I put in above. This is a very expensive process, and many people have been working on it. What happens to us is around 2010, at that time, these two guys here that are now professors, based Marty Waite is in France, and Farouk Morcos, University of Texas. They have this bright idea to show, although this problem is very complicated, the inference problem, if you're happy to do a mean field model into this problem, then these EIJs are just can just be computed as the inverse of the covariance matrix. This explodes this field, because that made these calculations much easier. It just became a big inversion, matrix inversion problem, instead of being a very expensive inference problem to do it.
Can I just ask very briefly, Jose?
Yes.
So here you're talking about the pair correlations. So I remember also from working on this a little bit myself. I guess there's this issue of the solvent accessible servant areas. So which can, because of the occlusion from these side chains, I guess you could potentially get these higher order interactions because of the solvent and the occlusion from other side chains. Is it an issue here or not so much?
It's always an issue, and I will comment on that as I go later into the process. Right. So here we are assuming that we can get these interactions. If you wait, I'll get to that, but let me bring this point. We assume that basically that's a good approximation into proteins, that effective pairwise interactions are good enough to predict fold. Although there are other interactions, you're going to see when I compute free energy later on. Okay, but for predicting structure, this assumption of just pairwise interaction is good enough, and you can do this mapping here. But I will get back to your point. Don't let me get away with it, okay? So if you can get now this covariance matrix, we can play the following game, and I can tell you now, I can collapse from this, I'm sorry. What happened? I can collapse all this information from the global distribution. I can collapse into trace the pairwise interactions. And now I can measure what we call DI. That's based P log P. Just tell me which pair of interactions of contact are more likely to happen. So from here, I can get the likelihood of which contacts are the most likely contacts to be on the structure, and that can help me a lot for prediction. Now, there are several interesting things that came from this calculation. Then now we can start to apply that for many different proteins that have many protein families. Normally, you need about of the order of 1,000 independent sequences in order to have good statistics to do that. That means this mass is sometimes much more powerful in bacteria. Sometimes we have more trouble in eukaryotes, but it has been very successful altogether. So here is a work that we did for 131 families of proteins. And this plot shows you how successful the method is. And you can see that basically you predict correct contacts almost very well for the first top or 20 or 30 contacts. If you look from this plot here, you see the difference just from our method DI and from mutual information. The red are true positives. You see, not only we predict better contacts, more contacts, but we also predict a more spread over the protein. So you can see with this information here, we are very good for a structural prediction. All this information that gives me enough in order to predict the structure of the system. Just out of curiosity, if I do the statistics of the top 30 contacts for 131 families, you observe that most of these contacts are about, on this peak, around 5 angstroms of distance between the C alpha atoms. This means that these are direct contacts. You have the second peak here that are the water separate contacts. But then you have the tail of the distribution. That in the beginning, people like to say, oh, this is just mistakes, you cannot be perfect. But I'm going to show you is actually very important. We are very curious about it. And we kept that for a long time. So how we did with this problem? So can we do with that information?
So just again, sorry, very briefly. So it's a direct coupling analysis. So it looks like the Kullback-Leibler divergence, what you wrote down. Isn't that effectively what the mutual information is, or how are they really different, these two approaches?
It's because you're getting the direct information. If you observe, the difference is if let me go back one case here for you. If I'm doing proof to information, I would just get this Pij that comes from Fij. That's the total pair probability distribution. Okay. I don't want to do that. I don't, because as I can tell you, the interactions can be direct between the residues or can be indirect via. Sure. So in order to get the direct information only, I have to get the global probability distribution.
Oh, okay.
Okay, and that's what I do using maximum entropy principle. And then I trace the pair.
Okay, I get it, yeah.
And that's what gives this difference. It actually does much, much better. That was the trick. That's something that started with Farouk and Martin Waite, and we did a lot of work on it, showing the advantage of going this way. Okay, so continue with that information. You observe the following thing. First of all, I want to show here's a contact map of a protein. Here I got one of this, 131 family, just got one as an example. This is the RAS protein. Here in shaded gray is the real crystal structure. In red here are my predictions that I call the correct predictions, and green are the wrong predictions. But if you observe, even the green dots are very, very close to the structure. So you can see you made very, very few mistakes. It's a very powerful method going all the way in terms of using to predict the structure. That became so powerful that when Joanna Sulkowska was in the lab, she was working mostly on proteins, but she decided to apply this method on the following idea. If I get proteins about 100 amino acids, and I assume I know the local structure, that means everything up to II plus 4, II plus 5, just the secondary structure, then just knowing the local structure plus the contacts that come from the I can predict structure with precision 1.5 angstrom. So the information that comes from this really help on protein structure prediction. You can see these are papers we have in 2011, 2012. That's when the methods are getting more developed. But now, as if you look AlphaFold, that people are talking a lot about it, covariance plays a major role on that. What they did, because they have more money than we do at Google, is they have bigger computers. They did a much larger database and very careful curation of the database for doing this work. But the physics is exactly what I told you. So that's, I think, is a lovely idea because it's something that comes from an entire tool from physics. We all knew it. We learned everything about Ising models and Potts models. And now we just bring all this battery that we learned from them, throw here, map into this problem, and now it's used by everybody. Okay, so it's a cool idea that comes from basic. There's enough information to sequence to bring you into structure. So as long as you have enough sequence, you do that. The other interesting thing about it is what I told you.
Sorry, can I just ask again about the AlphaFold? So isn't it somehow that they determine a potential, and I think that's what you did as well, but then don't they do the folding from scratch, right? So my question is, has AlphaFold told us anything about the different hypotheses about you had the folding funnel and then others had this kinetic control and so on. I didn't follow this now so carefully.
Let me tell you a couple of things about that I think nothing tells you directly, but tells you indirectly. The entire idea that covariance works is, as I told you before, is in total correlation with the idea of the funnel. So the idea of the funnel is that the native contacts have to be attractive and the non-natives have to be repulsive. So the fact that you're impulse the covariance between the native in the native context is because you have to have these compensatory mutations. Okay, so if the system was not funnel-like, then you wouldn't have this correlation between this sort of a covariance information and the structural information. That structural information comes from this idea of minimum frustration in the funnel that tells you these contacts have to be attracted, means they change in pairs. If one residue mutates, the second one has also to mutate in order to compensate it to do it. So what we got from here, what was very interesting is, if this is all correct and we believe the information is correlated, we are very happy with this part of the peak because it makes a lot of sense. These are the systems where I have larger DIs, right? The top couplings, here's for 131 families. They are actually a native context. But how about this tail of the distribution? I got very nervous with that tail of distribution. Say, this is not a mistake of the method. If you have a strong signal, you must have a reason for that. Right? Because these are things that the contexts are 10 to 25 angstroms apart, right? So they're not direct contacts. So is that just a mistake? For structural prediction, that's good enough. Because you have most of your contacts right here. So you're happy. But the question is why you're getting a strong signal down here. I say, maybe there's something more interesting here. And actually, what you observe is actually there's a lot of interesting things here. These things may not be contacts on your native crystal structure, but they may be contacts in a functional structure or when you form multimers, when you do dimers, trimers, or situational. So here's an example, for example, that we continue on that work. Here is for this protein, NRTC1. And we look, for example, one particular pair of residues here. They have a strong covariance signal. If you look on a single residue, they are very far apart in a single monomer. But if you look at the monomer interaction, they are pairs. So although they are very far on the single protein, when the multimers form, here's octomer, they are together on bringing the multimerization of these things. So that's very nice. So they're really pairs when the full structure is done. So it tells you the information on covariance not only help us in terms of understanding the structure of the protein, but also the interactions of the multimerization that comes here. So it's very cool because you come here, some of these residues, although they are here on the monomer, they are very close on the multimerization that they have. So this has become a big thing, not only to predict protein structures, but also to predict protein-protein interaction, particularly, as I'm going to tell you, about homodimers and homotrimers and situations like that. The other interesting thing is the following. Here's a case that we're very, very proud, and I think is one of the cases that really provoke the case. Let me get a particular protein that now doesn't have a single structure. It has two structures. On this case here, we are looking at this derived binding protein that's open before it binds and closes after the sugar is in there. So what do you observe from this protein? Just look at the top triangle here. The blue dots are all the contacts that comes from the crystal structure of the open structure. These red dots are the additional contact that comes when you get the closed structure. Now I'm going to compare this top triangle with what I get from my DCA, my DI information. And you observe there's a lot of correlation between these two parts. You also observe, and that's exactly like the blue one, you also observe a good correlation with the red part. These are the contacts that come afterward when it's closed. But you have this additional box of contacts that don't exist here. So when we do a model now, we do a model for this protein, we do a structural-based model where we include the possibility of all these contacts. And I let a simulation run, right? I put the energy term that include all these interactions. And now I cluster my possible structures. You see that you get the closed structure, the open structure, but you also get a twist structure. So in a sense, you observe that as this protein is closing, in order to bind the D-ribose, has also to twist. The D-ribose comes in and you retwist and close. This state has been predicted on some all-atom simulations, and some people are proposing experimentally, and we just get that from the data. So we are not predicting this state. Let's put it this way. We are just observing. Evolution has predicted that state, right? We are getting this from the sequence information, right? Our method is just extract this information from the sequence information. So as you observe, now we have this twisted state here. It's a very nice story and tells you the power of these methods coming together towards you. So let's continue a little bit more with this idea of this analysis. One thing we actually did when we moved into this problem was actually we didn't start on single proteins. We're more interested on dimers. This is a problem we are very interested in gene networks, this idea of histone kinase and response regulators, right? How these things do information into bacteria, and we're trying to predict these structures. So the first person to work on that was actually Alex Schulg, that's now in Germany. And Alex did the first structure prediction using DCA. The trouble is, if you have two different proteins, now you're not aligning a sequence. You have to align both sequences. So you have to create what you can see here, the super sequence. One protein plus the other protein, you have to align. You have to know which pairs come together. In bacteria, that's easy because the pairs that come together, they have the same operon. So you know which is supposed to go which, so you don't mix the response regulators with the kinase response regulators, but then you align the super sequences. Alex did the first paper on DCA in 2009. It's funny, a lot of people use this, but just to remind how that field started. When he predicted that structure, was our first paper, took us too much time playing around. So when we published it, came out together with the experimental paper, we could have published 6 months earlier, just not being aggressive enough. It's a great story that basically how DCA became the idea of being able to predict the structure. Here he only had, inference was much more expensive because we haven't done what we call the mean field DCA, but the problem came together. These days, Farouk and Ryan Sheng can do that for many, many pairs of proteins, interactions doing these things on pairs of proteins. So this mask are doing that. Now, the cheapest case is the homodimers. Because the homodimers, you go back to that plot I told you, you know the monomers, you just look at the tail. And now you look at that tail and say, which contacts of that tail involve residues on the surface of the proteins? Right? So if the residues are in the surface of the proteins here, then they are probably contact, and you use those surface residues that have large DI, large DCA to come together. And Ricardo Santos and Farouk working together, they show that for many, many dimers. You can predict dimers with precisions of the order of an angstrom or 2, right? And you can even predict structural domain swapping dimers using that information. So that's a very powerful idea helping you towards that goal. Okay, so that tells you about structural prediction, how much this method has changed structural prediction. So I'm going to use my next 15 minutes to actually tell you on more application that becomes interesting by using this method. There's lots of people using structural prediction. We're very happy about it. Let them use covariance. They have new inference methods, their entire field on covariance now. I'm glad we started that field, but actually there's lots of other things interesting you can do here. Actually, as you know, one of the big things in our field now is to work on genome architecture, try to find chromatin folding. And in chromatin folding, there's several gigantic proteins, what they call this SMC. That is this sort of structure proteins for chromosome, right? And there are cohesins, condensins. So the question is, can we predict the structure of these gigantic proteins here with the help of DCA? So if I get, for example, here, remember, these are the proteins that are responsible for making loops into this chromosome structure, okay? I won't have time to go into details on that. We have an entire field on that. I can give you an entire new talk on that, but that's not the point for today. The point here is just trying to predict the structure of that protein. So what we do here is the following. We get lots of these proteins, particularly in bacterial condensin, and we do the alignment, just like I told you guys. So what's cool here is we do the alignment of the sequence, and also, although these gigantic proteins don't have structures, crystallographers have been doing pieces of it. They have doing a little bit of their hinge. Let me go back one slide. Observe that these proteins have a hinge here. There are these big two coils that come together, and then they have these proteins down here where the motorization is what they call the glycines. Okay? So that's the nature of the structure of these proteins as you're having a look. So if I told you the crystallographers are able to find some pieces of the structure, and in orange here are the pieces that came from crystallography, like this piece of the hinge that is here, comes here, or some pieces around of the glycines that they have. So they have different pieces they cut together, they're able to crystallize. Now what we do is we apply DCA to this gigantic sequence and observe these are sequences of almost 3,000 amino acids, okay? So when we apply that to the protein, you observe that now from DCA, I can predict the entire structure of it. Even, for example, you see, you have two coils here, the two coil comes together, the structure comes here. But more interesting, I can understand the interaction between these proteins. So now I can start to look at this region. Here is one protein, here are the three glycines, as you can see, and here's the other protein. The first thing I look, there is two kinds of glycines. That's what they call SSP1 and SSP2. And actually, there was a question if you had what was the stoichiometry of the link, SSPA, and SSP2, and we showed that you have two SSPB and one SSPA. That's the best way of matching these results we have at this point when we run our structural-based model. So just to put here, then we use all this context and run a structural-based model. So what we see from here is something very cool. We can figure out the stoichiometry here. If I want to go back just a slide to show you, there was this question, if this thing was a single ring with two DNAs going through, or you are two rings of this protein with one DNA on each of them. The single ring required to have this stoichiometry, two SSPB and one SSPA, and our simulation showed that was the answer. The other thing here, for example, let's just look at one particular region here, the region around the hinge. Okay, so if I just want to look the region around the hinge, I'm just getting this piece of the protein and I look basically out of these interactions. You see just that region, that's where the hinge is, okay? What you observe as base, first of all, is like I told you, this tells you about the right stoichiometry. Now let's look the regions around the hinge. Here's where the hinge is. So if I look around the hinge, you observe that you have three boxes of DCA. This one and this one is exactly what agrees with the crystal structure. But we've got a second area of additional context. The beauty of that when we run our simulations now, when you do the molecular dynamics using Hamiltonian using these terms, is that now I show that this term here, this doesn't show the crystal structure, is actually open structure. The crystal structure is the closed structure. So actually this hinge can open and close. And that's how the DNA can come in and come out. So you see the power of this DCA of predicting this context. You actually can not only get information because all the pairs are important. And it's not the pair for just the crystal structure, it's the pair for all the important structures of this protein being put together. So what we see on this open structure, we can see now that basically you have all this positive charge on the surface that makes a lot of sense where they bind the DNA. And with this information in hand now, we are doing models. That's one of the big active research of what you're doing on how these models act. Basically, we start to now doing models, mixing all atoms force fields using the awesome model of Peter Wallens. So use our prediction using DCA as a starting model, and we get many structures for different cohesions and condensing, not only on bacteria, but also in humans, all different kinds of these proteins. And the nice thing now is the predictions are very good. I want to go in details with you. But the most interesting part is that you observe these structures, actually, they may not have a single isomers. They may have multiple isomers, depending on they can twist around them, because these motors are circular motors going around, twisting motors. And actually, we can show that, for example, on the case of cohesion in eukaryotes, there's some cross-linking experiments. You cannot explain the data with a single isomer. You have to do multiple twistings to show that basically, in order to explain the data, to tell you the problem these motors are rotating and twisting these things as they move around and try to make these loops into these proteins. So that creates one thing that's basically just for people to think about it. Should we think about these motors as rings, like handcuffs around these proteins, or should we think as braids? This sort of, they just move around the proteins, they move along, and that's a big discussion on how the motors work here. There's a lot of work on this area these days, and this is something that is a big discussion to tell you. Today, I just want to show the power of the method to bring it, to think that you're never going to get a crystal structure, but with the help of theory, being able to predict structures that large.
Just a quick question, Jose.
Sure.
So when you have the contact map, you can certainly, you make predictions for how the whole thing assembles, the stoichiometry, for instance. But how do you get these structures? These are molecular dynamic simulations?
Exactly. What we do is, like I told you before, we do a structural-based model, and we assume these interactions are attractive interaction in our structural-based model. So you create a Hamiltonian, where you have interactions between the amino acids that are attracted between the things that have larger DI.
Oh, you create your own,
Yes, we create our own Hamiltonian. Instead of doing, okay, I'm sorry, I was a bit confused on that, so let me be clear here. So what I do here is, in the past, I'm sorry, I'm very glad you asked this question. In the past, we get the protein structure, and you build a Hamiltonian when you made the native corrections attractive and non-native repulsive. That's structure. Now what we do is we use the same Hamiltonian, but we assume that the contacts that come from this DI, they're also contacts in our model. And then we can run the simulation. So that's how we get multiple structures from the simulation.
Oh, wow. It's really cool that you can make the connections and do molecular dynamics and improve that on top of that.
Exactly. And that's one thing that's actually becoming a very powerful method. See? And that's how you can get situations like here. That you can add the open structure, closed structure, but you run the simulation and you actually run a long simulation. And when you cluster all the results, you see that your results fall into two groups.
So one quick question. So to what molecular level or details do you capture the side chains? When you build your own Hamiltonian, is it a coarse-grained model with one?
Okay. So if you look we have been doing structural models for a lifetime, and we have a big data software server, SMOG2. And we can do both C-alpha level and all-atom level. Okay, normally what we do is we start the simulations at a C-alpha level just to get it, but then we do an all-atom structural-based model.
But then the force field, you're saying you don't use the standard force field, but rather you're using your own Hamiltonian.
My own Hamiltonian, and you can see how that is described over there. So in a sense, when we build my own Hamiltonian, when I have a full crystal structure, it's very easy to do an all-atom structural-based model. When you have this case here, when you have just interaction between the residues, you actually can only do a C-alpha level. So what we did here, as I'm going to tell you, just to repeat what myself, and I think it's a good point, we start to use our C-alpha model to get the basic structure. And then here with Peter, we actually run a full force field to optimize the structures, okay? So that's the strategy now. You get the initial big structure out of the C-alpha level, and then you add the side chains, and you run a full simulation with a real force field. Okay? But then just to going back to people here a little bit, I want to remind people, is that look at the size of this protein. That's about 40 nanometers protein, right? Base. Here's a nucleosome, just to give a size of scale, okay? To show how gigantic this thing is and the power of the method by putting all these things together. I'm not going to be able to do everything on the time given to me, but I want to do one more thing because I want to address what was asked before. And I think is Robert asked a great question before, is the following. Now, how about if I become greedy? By becoming greedy, I say, I'm not going to say that this DI is only giving me the information, but I'm also trying to say I can get the energy, because I'm going to tell you the following thing. When the contacts are formed, there are pair of residues that happen more often than other pairs of residues, right? And I'm going to tell you the pairs of residues that happens more often, they are more attractive than the pair of residues that happens less often. We can argue a little bit, there's some distortions of evolution here, but let's make that assumption correct. If you assume the assumption correct now, I can get the minus log of the probabilities and call these energies here as real energies. Here, I want to also go back to what Robert asked. Remember, these are just the pairwise energies, but let's assume they are sufficient. And if I assume that these energies are correct, I can now start to mutation studies in my system. Okay, and use the energy that comes from here. This is a result that we are very, very happy about it. This is delta delta G as a mutation study versus delta delta G experimental. You observe the correlation here. It's extremely good. I have here 6 different families of proteins, okay? Now, these results, each family is plotted separate from the other family. Like Robert said, you may have interactions about solvation around you, and you may have other terms that may be more complicated and pairwise. We are assuming that when you have mutation, most of these terms, how collapsed is the unfolded state, what's the solvent interaction with the outside, is not changing that much. So for the same family, the pairwise interaction is very good. When you put multiple families, it's more problematic, right? But it's amazing that for single families, how well we do it, how we correlate. Actually, we correlate better than most of the methods that are doing full calculations using real physical force fields. Now, doesn't mean, but like I said before, it's not that my calculation got better. This is result. I've just extracted information from the covariance data, right? We are getting this information. So to make this more interesting, what you observe here is that you can think about it that now sequence space is like evolution on sequence space. There's a temperature, what I call T-select. There's the temperature. When you make mutation sequence space, do you accept the next sequence or not? That's an ideal temperature, right? Of how you're accepting sequence on sequence space. This temperature is actually the slope of this curve here. Because remember, delta delta G from DCA is information theory. It doesn't have units. This one has units of energy. So this has to be multiplied by what we call KT-select to give the number. And KT-select is a number depending on the sequence between 150 and 300 Kelvin. Here's the case for the PDZ family. We are comparing the DCA prediction with this all-atom force field just for curiosity to show how well we do it. You see, if I just do the sequence that fold into the same structure, they have this minimum lowering energy, both in DCA and in the full force field. If I run this, if I now randomize these sequences, you see that you get what I call molten globule here, you lose information. So you see this very strong correlation between force fields and the things you get this T-select coming here for you. So that shows the power, how much is information is just on sequence information, because these families are evolving. So going back to what Robert asked, I want to remind that the fact that you have this correlation between sequence and structure is a direct evidence of the funnel. Right? The system is probing into this rough funnel. It's trying to look for structures going downhill, right? But you have the sequence that where you try to optimize with a little bit of roughness. So let's just conclude. So guys, how much time do I have now? I didn't pay attention.
I mean, you have, let's say 5 more minutes if that's okay.
Yeah, 5 more minutes just to show you one more interesting result on mutations. So I just want to show this result here because you guys are going to enjoy to look at it. Now, as I told you, in bacteria, you have this histone kinase that bind to these response regulators, right? And there, every bacteria, that's how their decision making is made, right? And you have about 100 to 1,000 partners in every protein. So what happens in bacteria? In bacteria, you have that basic, whatever is the signal from outside the metabolite, it binds here to a histone kinase. There is a phosphorylation. Now, you have to identify the correct response regulator. That's the beauty of it. These proteins are awfully similar. How can you differentiate between them? You have to identify this response regulator. Now, what happens in bacteria? The phosphorylation goes to response regulator, and this response regulator goes out and controls gene expression. But what is interesting is how the right kinase identified the right response regulator. So what we did here is we applied the same thing. We put the sequences of response regulators and kinases, right? And we try to align all of them, all possible alignments between these things in bacteria. I won't have time to go too much in details, but this is a graph that I really like. This is a result coming just looking at DIs on average between the pairs of residues at the interface of the binding interface, okay? If these things have perfect identification, okay? Here is a list, as you can see, of pairs of response regulators and kinases, okay? And if these were a perfect thing, you should have a full red diagonal, and everything else should be blank. You see that how the method here is actually able to identify diagonal is much stronger. There are very interesting questions to show you. There is a case of RSTA that's very wide here, this one here, because you don't have data. If you don't have data, there's nothing you can do. But the case you have data. And then there is the case of pairs like YFKA here, oh, I'm sorry, the RSTB here, that you observe there should be make pairs with RSTA, but this is RSTB is a very promiscuous response regulator, okay? So the method is sort of very, very nice and shows you on the spirit of these mutation studies that you can actually now start to identify pairs that bind together. This is results for E. coli. And to conclude, I want to show one result how this method can be done. I want to say a paper we published on molecular biology and evolution. This is, look on these response regulators just to show here, there is one of them that's FOQ and FOP, is one particular pair. On E. coli, I'll be focusing on it. These some experiments done on MATLAB's lab, okay? And what MATLAB did is these response regulators control the entire magnesium metabolism into your system, but pretty much what you're interested in is how this 4Q and 4P binds. What they have here is 4 residues. They're responsible for this binding, okay? And what Matt did, they mutate all these residues. So now that tells that they have 20 to the 4 sequences. So they have about 160,000 variants of this sequence. And what they did is, for all these sequences, say which ones is still active and which ones are not active. So for the experiment, they observed that about 1% of all these pairs is still active in bacteria, and about 99% is non-active. So the question is, can we use our method to design? That means, in this case, can we identify the good ones and the bad ones? using just GCA, okay? They have all this information. So what we did is the following thing. We came into our system, and again, we got for Q and for P, and we identify all possible pairs. Okay? If I just do for Q and for P, and I figure out which are the good ones, I can look at that my method. If I look at my tail, I identify only good ones. Okay? Now let me tell you what I mean by that. Here we are not saying they are better, they're just saying if they're active or not active. But the question is, if you think about design, is can I use the tail of my distribution to get this? So what I look here is if I get my top variance, observe that I have an 80% prediction power. What's amazing, if you think about it, that only 1% is positive. So the method is very good. But it's even more interesting. If I put an extra screen that tells you when I do the mutation, not only I want to say I'm doing well, I want to make sure that these mutations will make me for Q to bind to another pair that's even better. So the question here now going back, when you do this design, you not only want to make a better binder to the response regulator that you want, so you want to increase specificity for the one you want to bind, but you want to decrease the specificity for the ones you're competing against, right? Because if I look back on this game here, this is a competition game. You just have to identify yours better than the other ones. So when I come here now and I tell you, not only I'm going to get good binders, but I'm also going to remove if I bind better to someone else, then look at here for my top 50, I have 100% prediction power. Tells you a very interesting story about pushing this method forward. I'm going to stop here, but I just want to say a couple things before I finish. I won't have time to tell you about how these tools have been used in chromatin structure prediction because it's just really there's not enough time. But if you want to have a look, it's very interesting how we have been using this data just to tell you the notch delta, the same tools in a complete different way. Just use the mathematics of it to be able to predict from ChIP-Seq data to chromatin types. Again, by using covariance. So I'm just going to leave that just as a taste with no explanation. And I want to finish here by thanking our collaborators. And we always have been getting a lot of support from the National Science Foundation. But as you can see here, the two bottom things, the Welsh and the ChIP-Seq, is clear evidence you are in Texas now. Okay? Thank you very much for your attention.
Thank you so much, Jose. Fascinating talk. I mean, this is basically covering part of your career at Rice, as I can tell. And then you didn't even talk about, you know, as you say in the end here about the chromatin folding. So I guess it's a completely additional area you worked in. And
maybe I have a chance to tell you that story at some point. It's a beautiful story also.
Yeah. And then you did also, I guess, cancer research. Is it right?
Yeah, that's correct. We have been doing a lot of work on epithelial mesenchymal transition and metabolism in cancer. So there's a big effort on that direction. I have been fortunate of having a fantastic group of postdocs that do around it. So basically, I have the luxury of giving the talk under what I call the Imperial we. We say we did that, but actually they did that. So I want to thank all these great people going around. And I think this work, like I told you, a lot of this work, there are 3 people involved. I told you about it. Basically, the 3 big things here, it was Farouk Marcus that I told you, Martin Waite, that's now in France, did the early stages, and Ryan Shang, that's still a postdoc in the group, did all this last part in terms of these response regulators and kinase parts. So this is basically a group that 3 postdocs actually maneuvered this entire group with some graduate students, but they are the main ones doing this work.
Yeah, it looks also like a fantastic collaboration with Peter Wollins and Herbie Levine.
Yes, that has been, like you said, you saw Peter there. Peter and I have been together for long. The center of basically, I also want to tell people that you guys are welcome to visit us here in Houston. I didn't tell you that. I want to say that, let me just stop sharing here. Maybe I.
Okay. Yeah, so just a reminder for people to submit questions if there's anything. Otherwise, I keep going. There's so much in this talk.
So I have a quick question. So I found that the idea of matching your information entropy to some sort of real free energy very interesting, and you came up with some temperature to relate the two. So can you say a bit more about that? Can we read more into it?
That's an idea. That the idea of selection temperature is an idea that actually started a long time ago by Grossberg and all these people. And with DCA, we actually have made it concrete. And that's a beautiful physics question. So you can imagine that basically, as you have, okay, let me tell a couple of things here, just say, we are using all the data from evolution. There are a lot of people that basically now they're trying to look at data together with evolution trees and see how this data work. So there are lots of things to do there. But here is the idea that as the sequences evolve in evolution, we know that basically the way evolution works is you live or die, right? That's how it works. But we are trying to make a simplified version of evolution. Our idea is this live or die may be associated to the stability of the protein. That may not be 100% correct, but that's pretty good. Okay, if that's true, you can imagine that as you evolve on sequence space, now you're going to have like a sort of evolution Monte Carlo, which sequences are accepted, right? So basically, so your T-select tells you in a kind of Monte Carlo view, if I go uphill, if my sequence less stable, is that going to be accepted or not? And that's what T-select is. So it gives a temperature, not a real temperature, but more a sort of an effective temperature in space that do that. What's very nice is there's sort of theoretical calculations, but we are associating T-select by saying the following way. If you looked at plot I show you between delta G that we get, our case is just information theory. We have to multiply by energy to get a real, and I compare that with experimental or a force. That temperature, that slope of that plot gives you that temperature. And that temperature is associated with what we call the selection temperature. And then we show that actually is consistent. We have lots of other calculations supporting that evidence. But it's a very interesting thing to be discussed in terms of thinking about how sequences are accepted and rejected as we evolve on evolution. And we are using that from RMS to do that. And it's sort of a cool idea that comes there. Grossberg put before, but now we can actually formalize, and you can see that basically that they are related.
Yeah, so I think it's very nice to relate the two. I guess my question is 126 Kelvin. Does it mean anything? Can you relate it to anything? In that this is your it's a real Hamiltonian.
It's a very good question. That's something. If you read our paper, we are very agnostic about it because we couldn't resolve. Okay, for some families it was at 126 Kelvin. For some families are 250 Kelvin. Okay. The cool part is that you get temperatures on the hundreds of Kelvin. You don't get anything that's 1,000 or 1. But it is a very interesting story. How would I relate that? Now, if you allow me to speculate, because the speculation we make now, if I was the reviewer, I would reject the paper. But since I don't have a reviewer now, I can speculate. You could expect that proteins that are more robust, that can tolerate more abuse, they would be a larger T-select. They would be more flexible. As you do mutation, they will say, okay, I'm allowed to explore more because I'm able to survive in a better way. And systems that are very sensitive with a very minor stability, they may not survive exploring that landscape that much. Now, we have some evidence for that, but that's very, very speculative. Okay? Actually, Ryan Shang is working to see, can we actually have enough data that the statement I told you I can make in a quantitative way, right? Not more in a folkloric way the way I'm presenting here, right? So it's like, it's not something you want to show by showing an evidence. One protein works one way and the other works the other way. You want to have a bulk of proteins and look at data. And I think we believe we can do that by staying on bacteria, where there will be enough protein and enough sequences. If you go to eukaryotes, it will be very complicated, but just look evolution in bacteria. If we can get enough information, we could answer this question. But that's the feeling that we have right now, and appears to be consistent with the small amount of data that we have.
Yeah, but you've just generalizing your speculation. So if you look at organisms that live in a colder temperature and compared to organisms living in hotter temperature, would you expect a T-select to be different?
So you see, you are making the question more and more difficult to me, and you are right to make the question more and more difficult for two reasons. I just rather get a protein like myoglobin and cytochrome C. They're tough birds. That's why physicists like to work with them. And notice they have a higher temperature, and other proteins that are involved in very detailed signaling and do these things. The high and cold temperature is a much more sensitive, discrete aspect they have on that. The other thing that you have to be very careful as you run DCA and DI, I was a little bit cavalier about it during my talk, is you have to make sure you have enough data. For all the sequence, you have to make sure you have enough sequences. So that's another thing why, for example, when we start to put evolutionary trees and try to break into pieces, your data start to dilute. Even when I start to mutation states, I'm already sort of, when I get just pure DI, I will have lots of sequence, right? But when I start to split the data, then in bacteria you do well. So all these cases, you actually have to be very careful to make sure you have enough data.
Thank you.
Now, on top of it, just to be curious here, remember, I'm assuming all this statistical aspect of sequence in a sort of a statistical mechanics distribution, right? Basically, remember, I could have accidents of evolution that avoid some sequence to exist and not exist. So you have to think about that my entire landscape of sequence is whatever evolution gives to me, right? So it could be in principle that there were other sequences that do that, we don't have it. That's not a problem on the funnel aspect, okay? Because the funnel aspect doesn't have to tell you that's the absolute minimum of everything. Just have to see is the lowest minimum that's commonly reached, right? So it could be that you just live in a universe of certain sequence, that's fine, both kinetic and thermodynamic, as long as that's the only area you can explore during the entire process.
So when you increase the temperature to 300 Kelvin or something, factor 2, 3 higher, would you get unfolding in these coarse-grained simulations, or is it minor errors you get?
Oh, okay, no. Our coarse-grained simulations have been, okay, our coarse-grained simulations, if you look back, now let's get the folding situation. They are always calibrated around TF, okay? So normally what we show for these cases is, that's a beautiful thing that happens in the system, and we can go over loads of paper of discussion. The interactions between residues in that Loci alpha model, they tend to be of the order of KT. Actually, in order to optimize data, they are actually, if epsilon, the interaction between residues attractive is 1, KT is around 0.6, 0.7. That's a scale for us that for most proteins agrees to give the folding temperature of the protein in the units of the energy we have, right? So on the structural based models, you always have this to go there. The beautiful part about it is that this series of parameters that we have appear to work for most proteins. That's the power of the structural based models. Now, going back to what Robert asked, and just for your curiosity, because you asked before, if I run a structural based model, I get the geometry. We say that since the paper we published on ChIP-Seq in 1990, it's very important for you to keep in mind, is that you get all the right geometry states but the barriers between the states tend to be low. That's a nature, right? You need higher order terms to get the barrier correct. So that's the interesting thing about pairwise, and we have done that basically. If you look the work of Paul Whitford, that's now Northeastern, my postdoc, he did a lot of things by using a structural-based model to get the full landscape, and then you do all random simulation to look the transition between the local states. You cannot do the full system with all force fields, but you can find all the basic basins, and now do just transition between those basins. And these are not an interesting thing to do it. We are very good about getting the stability energies and get the right geometry, but the barriers tend to be a little bit lower because the barriers tend to have these. So although these higher order terms are not necessary for a structural prediction or to find geometrical features or transition states, they tend to underestimate barriers.
Mm-hmm. Okay. Yes, thank you so much, Jose, for all the time you spent with us. It was a fantastic talk. It's maybe a good time to conclude this event. Yeah, it was really fantastic to have you here. So thank you again.